Look up at these surgical lights. The moment the IV anesthesia enters your bloodstream, it takes just seconds for the room to blur and fade to absolute black. Your consciousness simply drops off a cliff. Looking at this patient on the table, we see a strange gap in biological understanding. The heart continues to beat under the drapes, the lungs steadily pull oxygen through the mask, and physical reflexes remain active, but the conscious you is completely absent. Classical biology treats the human brain as an incredibly complex electrical grid. The problem is, anesthetic drugs do not completely stop the brain's electrical activity. The biological hardware is still running, and the wiring is still live. If the electrical network remains firing while the subjective mind is shut off, then consciousness appears to operate on a level that classical electrical signals cannot fully explain. This is Oxford mathematician Roger Penrose. In the 1990s, he partnered with an anesthesiologist named Stuart Hameroff to investigate whether a deeper, subatomic process was responsible for the mind. Working within these halls of Oxford, their collaboration forced an overlap between two totally distinct fields of science, clinical medicine and quantum physics. Together, they proposed that consciousness is a result of quantum physics phenomena occurring at the subatomic level. Watch what happens when we zoom deep inside a single brain cell. Past the outer membrane and the biological machinery, we find a microscopic network of tiny glowing cylinders known as microtubules. Penrose and Hameroff argued that to understand the mind, we must look at the brain as a biological quantum computer. Looking closely at this microtubule, it's 3,000 times thinner than a human hair. Its walls are built from tubulin proteins stacked like bricks. Notice how they blur. At the atomic scale, a single molecule holds two different shapes simultaneously, a state known as quantum superposition. In the macroscopic world we live in, that is impossible. A door is either open or closed. It cannot occupy both positions simultaneously. While these millions of tubes provide a structural skeleton for the cell, they also function as billions of subatomic switches, processing information through these overlapping quantum states. Physicists initially rejected this idea. Maintaining fragile quantum states in a laboratory requires heavy lead shielding, and temperatures dropped to almost absolute zero to prevent interference. The human brain is a noisy, chaotic environment, running at a warm 37 degrees Celsius. Expecting a delicate quantum reaction to survive in this heat is like expecting a penguin colony to survive in the middle of the Saharan desert. But look at the water molecules inside the tube. The protein walls force them to snap into a perfectly ordered crystalline grid. This acts as physical armor, deflecting thermal noise and keeping the fragile tubulin superposition intact inside. Here at the UC Santa Barbara campus, physicist Matthew Fisher published work in 2015 showing how the internal spin of phosphorus atoms might similarly hold protected quantum data at biological temperatures. Through millions of years of evolution, biology engineered a protective casing that allows a warm quantum computer to run smoothly within the human body. By 2009, laboratory researchers had isolated microtubules and observed quantum behaviors directly. These findings provide a new perspective on the mechanism of anesthesia. Watch as an anesthetic molecule floods the system. It chemically binds to the tubulin protein, shattering the aligned water crystal into chaos. With the armor gone, ambient brain heat floods in, forcing the dual-state superposition to collapse into a single, static position. This suggests anesthesia works by disrupting the protective geometry inside your brain cells, unplugs the quantum computer, and stops the process of consciousness. This subatomic activity offers a potential explanation for the binding problem in neuroscience. When you look at a flower, its color, scent, and texture are processed in physically separate regions of the brain, yet they arrive in your mind as a single, unified experience. Classical chemical and electrical impulses move at speeds that struggle to explain how this data is synchronized so perfectly. But mapped out here, we see a network running on quantum entanglement. Distant microtubules can form an instantaneous link across the brain, sharing data with zero latency across separate physical regions. This hypothesis suggests that our seamless perception of reality is possible because the brain utilizes connections that operate outside the classical limits of time and space. Artificial intelligence systems are limited by classical computing. They process logic sequentially, using a binary language of ones and zeros. Because microtubules utilize superposition, they can evaluate many probabilities simultaneously, providing a biological basis for human intuition and creativity. Penrose argues that consciousness is an inevitable result of the universe's quantum structure, a phenomenon governed by the same fundamental laws that move atoms. Right now, as you process the concepts in this video, millions of microscopic tubes inside your skull are executing a precise quantum dance, a moment of wonder. By facilitating the very act of comprehension, this internal network remains the most complex information processing system yet discovered in the known universe.